In this video, we will learn about northern blot analysis. Northern blotting is a molecular biological method used to study RNA levels. This is done by detecting specific RNA molecules in a mixture of RNA. The detection is based on hybridization between RNA and a label probe. Northern blots are used to determine the identity, quantity, and size of specific RNA. For example, it can be used to analyze the abundance of RNA during different stages in cell or tissues. It can be also used to determine the expression levels of the specific gene under different experimental conditions. Here you can see the workflow of the northern blotting. The protocol begins with the extraction of total RNA from cells. Then, the RNAs are separated according to size using agarose gel electrophoresis. Following separation, RNA is transferred from the gel onto a nylon membrane. This is done by capillary transfer. Once the transfer is complete, the blotting membrane carries RNA bands retaining the same separation pattern. Next, the membrane is incubated with a small piece of oligonucleotide named a label probe. The probe has a sequence complementary to the RNA we want to detect. This facilitates hybridization with specific RNA molecules in the sample. In addition, the probe contains a label used for detection as the final step. In the next slides, we will go into more detail through the steps of northern blotting. Let's see the first step, the isolation of total RNA. The basic principle of protocols for RNA isolation starts with lysing the cells by disrupting the cell wall and the membrane. This is followed by the separation of RNA and the removal of the DNA and proteins. Several RNA preparation methods are available, and here we will mention two: the organic extraction method and the spin column-based method. Organic extraction methods are common for RNA isolation. During this process, the sample is homogenized in a phenol-containing solution and then centrifuged. During the centrifugation, the sample separates into distinct phases, and the process is also pH dependent. These are the lower organic phase and the interphase, containing denaturated proteins and genomic DNA, and an upper aqueous phase containing RNA. The aqueous phase is carefully removed, and RNA is collected by alcohol precipitation and rehydration. Nowadays, RNA isolation is done using commercially available kits based on membrane filters. These utilize membranes that contain silica or glass fiber to bind nucleic acid and are placed in spin columns. Samples are lysed in a buffer containing RNase inhibitors. Then, the lysates are passed through the membrane using centrifugation. This results in the binding of nucleic acid to the membrane. An additional step of DNase treatment is applied to remove the genomic DNA from the sample. The membrane is then washed to remove impurities. Then, RNA is eluted with RNase-free water. After the isolation, the concentration and purity of RNA are determined by a nanodrop spectrophotometer. This is done by measuring the absorbance at 260, 280, and 230 nanometers. Common problem during RNA isolation is the presence of ribonucleases or RNases. These enzymes are extremely stable, universally present, and generally do not require cofactors for their activities. The presence of RNases during the RNA preparation can degrade the RNA, leading to the loss of sample. To avoid problems with RNases, purification procedures should take place in the presence of RNase inhibitory agents. Typically, these are strong denaturation agents such as guanidine salts, sodium dodecyl sulfate, or phenol-based compounds. The commonly one used is sodium dodecyl sulfate solution. Here are also some tips for working with RNA. It is important to wear gloves, use sterile fresh tips and epis. Sodium dodecyl sulfate solution should be used to clean the work surface, all glassware and plastic parts. And finally, make sure to separate RNA isolation from other experiments. The next step is the separation of RNA according to the size by the gel electrophoresis. For the gel electrophoresis, we can use native agarose gels or denaturing gels containing formaldehyde. Alternatively, the sample buffer contains formaldehyde. Formaldehyde contains a carbonyl group that reacts with the imino or amino groups of nucleotide bases. This prevents normal base pairing and maintains the RNA in a denaturated state. It is often used since RNA can form extensive secondary structures via intramolecular base pairing. On the agarose gel, we can evaluate the integrity and overall quality of the total RNA isolated. This is done by analyzing two distinguishable bands corresponding to 23S and 16S ribosomal RNA. Further on, we continue with the step of capillary transfer. During this step, negatively charged RNA is transferred to a positively charged nylon membrane. This is facilitated by capillary transfer. The usual structure of the blot is depicted in the scheme. On the bottom is blotting paper having ends soaked into the transfer buffer. On top of the paper, we place the agarose gel facing upside down. Then, the nylon membrane is placed on the surface of the gel while avoiding any air bubbles formation. A few more filter papers are placed on top of the membrane. This will help with the capillary transfer. A glass plate is placed on top of the blot to hold everything in place. It is also recommended to put some weight on the top. The blot is left overnight to obtain an effective transfer. For the hybridization step, we need a short oleonucleotide called a probe. The probe has a sequence complementary to the RNA we want to detect. Additionally, the probe is labeled to facilitate the detection. Traditionally, probes were labeled with radioisotopes. However, non-radioactive alternative methods for probing neutron blots have become popular. A commonly used non-radioactive detection method is using the probe labeled with deoxygenin. The probe is then detected using an anti-deoxygenin antibody. Deoxygenin labeled probes are prepared by PCR amplification. During the reaction, deoxygenin conjugated deoxyuridine triphosphate is incorporated into the newly synthesized probe. In this nucleotide, DUTP is linked to the steroid hapten deoxygenin via a spacer. The PCR results in the generation of a specific and labeled probe. To ensure the probe specificity, it should be at least 25 bases long. After the capillary transfer, RNA molecules are immobilized on the membrane through covalent linkage. The linkage is achieved by exposing the membrane to UV light or by heating it to around 80 degrees Celsius. It is crucial to immobilize RNA molecules to avoid losing them during the subsequent processing steps. Prehybridization, or blocking, is required before probe hybridization. This prevents the probe from coating the membrane. Good blocking is necessary to minimize background problems. The hybridization step allows for the identification of RNA blotted on the membrane with a labeled probe. In the process of hybridization, a tailored probe binds specifically to the RNA of interest through base pairing interactions. The hybridization forms the basis for the detection of RNA. The specificity of hybridization between the probe and the RNA results in the accurate identification of the RNA segments. The base pairing between a labeled sample and the fixed RNA is ensured by complementary hydrogen bonds between the nucleic acids. At the molecular level, hybridization begins as soon as the ends of two complementary molecules come together. Once the pairing of bases is established at one end, it proceeds in a zipper-like fashion throughout the length of the molecule. Factors that influence the efficiency and specificity hybridizations are length, GC content, and number of mismatches. For example, longer hybrids have higher total binding energy and stability. GC-rich hybrids are more stable than AT-rich hybrids. The total binding energy of mismatched hybrids is reduced, making them less stable. After the hybridization, the membrane is washed to ensure that the probe has bound specifically, preventing background signals. After this step, the membrane is ready for detection. The last step of the Norton blotting is the detection. After hybridization of the probe to the target RNA, the resulting hybrids are detected by an enzyme-linked immunosorbent assay. In this example, we use an antibody to deoxygenin conjugated to alkaline phosphatase. On the blot, we colorimetrically detect products formed by the alkaline phosphatase activity. The substrate is 5-bromo-4-chloro-3-indolyl phosphate used in conjunction with nitrobutyl tetrazolium. Once hydrolyzed by alkaline phosphatase, the intermediate reacts with MBT, producing an insoluble precipitate that is dark blue to purple in color.